class, and I know I'm a little bit behind on a few things, so I know you have the answers to the energetics problems, the first ones we did, and if you're having trouble seeing that, the only trouble I think people have had when they wrote to me is they forget to put the slides on presentation mode, and if you hit it, then a little square or something moves away, you can see the answers. So, but I gotta get those graded and get your grades, so I know that. And then we have an assignment today, a discussion, and then you've been doing quizzes, and don't forget your supplementary quiz. Get that done before next week. If you haven't done that and you kind of forgot about it, jump on that, okay? You want to get those points, and also make sure you know about genomics. If we do that, we're pretty much up to date then, right? Actually, I have a few things in here. Yeah? No? Okay, well, that's where I think I'm at. I always have nightmares about losing assignments and stuff, so I try to keep everything together. So, okay, the assignment for today, some people come and talk to me, a lot of people have written to me, sent hate mail, slashed my tires, things like that, but I'm bigger than all that. So I think people are doing pretty good from what I can tell, just from what people are telling me, that the first one, you looked at the reduction potential, and you're looking at the carriers that fall within that. Some of you may have thought, I wonder if there's a part in the book on advanced metabolism that covers that. You know, that'd be a way of looking at it too. The book kind of tells you some answers. That's what books do. The third one is tricky in the way it is written, but it's truthful. It's medicine, right? Yeah, medicine I went through, we read over and over again. I wanted to make sure, because when I read it, it made sense, but sometimes when you start dissecting it with somebody, you know, it doesn't. But, you know, the fermentation is telling you about the condition, but then it flips over into respiration, and that's the key to that one. I don't know that anyone had a lot of problem with that one. The middle one on culturing is tough. Okay, there's stuff you know, and there's stuff you're going to learn, and I kind of pushed you with that one. Okay, so at very least, you know, think about a compound you want to degrade, the conditions that best degrade it to the best of your knowledge, and then what other stuff you're going to add to the tube. You know, you're just going to add your pollutant, or you're going to add something else. Well, look at nutrition. What would you do? You know, I know you probably won't get down to the millimole of everything, or maybe you will, but chances are you won't. But you can tell me what kind of stuff you're going to do, and then how, you know, what is your objective? You know, trying to get a microbe out of, you know, we're going to get your inoculant, you know, and then what microbe is going to degrade your thing the best? How are you going to find out? Maybe you're discovering a new microbe, which is totally within the realm of possibility. I mean, you guys could, if you put your mind to it, go out and start naming and finding new species. That's actually something you could do should you wish to. You can name it after yourself. You can name it after your mom. You know, that'd be kind of cool. Maybe. Anyway, look at that. If you turn it in and you're having second thoughts, or you want to do an addendum, you can add stuff or talk to me about it. But I'm looking also at the effort and your rationale. If we don't push ourselves beyond, you know, just, okay, memorize carbon, hydrogen, oxygen, you know, I mean, you know, stuff we kind of know. We don't push ourselves to understand the science, and this is a way of thinking. You know, you always say critical thinking. A lot of people don't know what that means. You know, I think it's thinking outside the box. This is the box. You know, this is thinking about the box and what goes in the box, and this is thinking through and combining things. So don't the underlying message. Don't, you know, sell your house and move away, and you know, don't get too shook up about things. On the other hand, you know, apply yourself. You're a third year. Well, it's a third year class. You may be third, second year, whatever. But you know, this is a little bit more than the basic meat and potatoes, and give it a good shot. Okay, that's what we're looking at. And if you're still upset, you can still come and talk to me. Or, you know, I have no problem talking about microbiology with you. Okay, next week, exam, yeah. Thursday, and people in lab, it's kind of double on you, but we have a Tuesday lab, so kind of pace yourself. I try not to put the exam on the same day, although 2 exams a day is acceptable on the university level, but I'd rather avoid it. Anything else I should be knowing about or you need to know about? Get a review sheet up. I have one. I just keep forgetting to do it. People ring me, where's the review sheet? Okay. Well, they're being very nice about that, actually. But, you know, in other words, get me gear on me. I'm okay with that. I'll do it. Anything else? Okay, one little change. If you're reading ahead, I'm hoping that you've only just started going into that genomics chapter on 6. Every year when I present the genomics the way the chapter does, it doesn't feel right to me. I really feel, and this is the way I present the course, I really feel that we should stick with metabolism and put that one behind us before we get into the genomics evolution and everything that's going to lead into the diversity of organisms. So, I would like to go into advanced metabolism, a very short portion of that. I mean, advanced metabolism is its own course. But finish that up to give you a hint of some of these different trophic stages we're talking about, and leave genomics to when we get into evolution, and we talk about genetic engineering and all this stuff. That's a good time to talk about genes all at once, is what I'm thinking. Anyone have a problem with that, or really upset? I mean, if you started reading it, we'll still get to it. It's just a matter of timing. I think keeping concentrated is a good idea. Well, let me know what you think. That's what I'm thinking. It's going to be better. Well, we're going to talk about growth. I think we can get through most of the stuff in here. I don't think, again, I don't think this is going to be anything you're going to run out and go, oh my God, I never thought about that before. I think we have it. This is probably where we left things off. Well, I know this is where we left things off, but this is probably some of the most new stuff to us, to think about how cells actually make more cells of themselves if you're starting from one cell. It's different if you're dividing and making more cells, like, you know, you were a little zygote at one point, a single cell. Now look at you, you got a trillion cells and all kinds of differentiation. It's great. Well, you know, how do you make more of yourself if you are, if you are only yourself? And then we talked about the replication, the divisosome, and we talked about these different proteins. And I think one thing I might have, I was looking over the lecture this, before coming here, and the one thing that's going on is that you have autolysins in here at the FTS-Z ring, and they're actually dissolving the peptidoglycan in a small portion, so you can still keep some kind of tension and integrity in that cell. They're not just dissolving the whole cell. But that's what's kind of interesting about this, kind of dissolve and unweave this thick peptidoglycan in order to weave it back together again. And so then you have this wall junction where the peptidoglycan is being then wall bands that are being reformed. And this is confusing. The division's already happened here in this one. This must be, I don't know for sure, I'm guessing it's Streptococcus is dividing on the ends, and that's how you get chains, as opposed to sometimes they'll divide on a different plane, and you'll get tetrads, Sarcina, or Staphylococcus. I mean, you know, it's the division plane that is defining of arrangement. Okay, but here we have this, and we can see the you know, where the Z, the septum, where the Z ring is. You can see these wall bands where things are being kind of growing back in and over it. So I don't want to, you know, flog this horse to death, but it is kind of a fascinating process. And then we talked about what goes in to the peptidoglycan. We have a bactoprinol. We've seen the idea of prinols before. And just that idea as a carrier, I'm not, so if you recognize that word with the way the monomer shuttles that in, that's good. I'm not going to ask you for the structure. I mean, you don't need to know that for this course. So the last part of it, I'd like you to know this term. It's important. We talked about it a little bit when we talked about the cell wall, but let's look at it again. Transpeptidation, the final step in cell wall synthesis, you bet, forms a peptide cross-links between muramic acid. We know that. Okay, when we look through the cell structure, go back and look at muramidase and NAC. glucosamine, and then, well, we talk about penicillin. So this is where we have these peptides. They can be variable by species, so I can't really say, well, this is how they always look. They can, and we had a few slides earlier that showed there's variability in there. We have, some unique amino acids in there, usually DAP. That's not that big of a deal for me, but the point is that they'll make this nice bridge across between strands. So it's kind of making this weave in the analogy I was making with us. So that has to happen if you're dissolving your peptidoglycan. You got to weave it back together again. So it tends to make sense to us, and so we go on. Okay, I think we talked about this. So let's look at this slide. We're going to talk about growth now. Growth as the idea of increasing numbers, and it starts out pretty simple. Primarily, we're talking about binary fission. We're not going to talk about mix of spores and all that stuff now. Okay, so we go with binary fission. We're on a log base kind of situation, and growth is exponential. And this just shows kind of what we know, but there's a few little things in here that captures our attention. Okay, I mean, now here's numbers of cells, logarithmic. So of course, that's going to straighten out this exponential curve here. And we have time and hours. So this is our progress of our growth curve. And I don't think, that's too surprising. Now, we have a cell here that is, this could be E. coli in a culture at 35 degrees. E. coli can reproduce as fast as 20 minutes. So that's a good one to think of. On your stomach or in your intestines, it's probably not that fast. But, okay, fair enough. Who cares what it is? What are we getting? So every half hour, this particular culture is able to double. So 1 to 2, okay, starts out slow. Okay, it's, not too much. Okay, 1 to 2, 2 to 4, 4 to 8, start getting, and all of a sudden the numbers start to kind of escalate. Okay, again, not a surprise, thanks for that, but at the time when we have 20 regenerations, okay, 10 times 2, we have 1 million cells. Okay, we've had, 19 regenerations, 19 generations. First one wasn't counted, of course, I was wrong on that. So 19 generations, we have 1 million cells. Okay, that's in 10 hours. So when I hand somebody a culture after 24 hours growth, everything's growing in exponential growth. It's still doing this. Okay, and we'll talk about growth curves in a second. That'll make more sense. Okay, but in 24 hours, we're sitting in 10 to the 13th, 10 to the 15th cells per mil. That's an incredible amount. That means like a drop of coli culture, if it falls on your hands, you've got millions of bacteria there now. And then you go like, well, I see why you want me to wear a lab coat, maybe some shoes that, I might have to autoclave or throw out if I drop a culture on. I get what you're going at. That's a lot of organisms. If we had 1 million cats in this room, it would be filled up. I mean, it's just crazy how much organisms are sitting there. So not all cells grow the same. Okay, we get this idea that, they're bacteria, they're all the same. They're everywhere. No, bacteria aren't everywhere. Okay, some people write that down. There's places bacteria aren't. Probably the inside of my autoclave doesn't have as many bacteria, I'm hoping. Okay, and we think they all grow fast. No, that's just wrong. As you're starting to appreciate, each population, each species does things a little bit differently, which is good for us because we want to talk about different species, but also we start to learn some of the characteristics. So first part we've covered, growth curve, n equals the original number times 2 to the power of n. Okay, that's really how we develop these growth curves. If you're measuring, you can work backwards on that if you know the generation times. Either way, let's just look at the graphs here. In our top graph, we have our time goes to 6 hours, but they're both basically on the same time scale. And then we have essentially abundance going on the y scale. So that's easy to see. So now we see two different slopes. We see a slope of 0.05, okay, it gives it to us. Of course, you guys can calculate that, that's not a problem. And a slope of 0.15, okay. You could either backtrack, you know, and figure out your number of generation times using G, and here it becomes pretty transparent. You see that G is basically, you know, T in these cases. So, but you could backtrack, that depends on what you're measuring. So that's all pretty good. So, but what do we notice? Okay, well, we know that the higher the slope, the higher the rate. We've learned that since 6th grade or whatever. So we know these guys are multiplying faster. So within 4 hours, we're sitting at 8 times 10 to the 7th. 4 hours here, we're about 1/4 of that. Okay. So we plot these things out. In lab, we're looking at how many cells are in the turbidimeter, a spectrophotometer. Cells not only grow at a different rate, they give different readings when they scatter light, and that'll become important in a slide or two. So basically, if you're studying, this might be important to you. In one of the examples I gave you, I put spectrophotometer absorbance against time, and you saw growth rates, all right? That was there just as an example. Some people said, do I have to explain that? No, I was just trying to show you how people use specs to look at progress and growth. You'd want to know what your cells do normally, you know, that's called a control, right? Before you start adding different, you know, electron acceptors, different carbon, you know, so knowing your growth rates to begin with is fairly important. Okay, the growth curve. If you haven't, probably saw this before, so if not, pay attention. If you have, here's a review. We have 4 stages, and it's worthwhile just spending some time talking about. We have a lag phase, an exponential phase, a stationary phase, and then a death phase, okay? What we're talking about here, though, is batch culture. That is, you're inoculating some bacteria into a test tube or a flask. You know, some people say, well, it's still an open system, you know, the air can get in and out. Yeah, but you're not adding more nutrients, you're not taking away wastes. In a moment, we'll talk about alternate ways of batch, alternate ways of culturing. The other one is a flow-through system, what they call a chemostat. You're a chemostat. You're taking in Pop-Tarts and so other things, you're eating things, oatmeal or whatever you brought today. And, you know, that's fueling more things, and you're getting rid of other things. I mean, like it or not, they're going out. So you're like a continuous culture. You know, you probably never thought of yourself that way. Hey, Mom, I'm a continuous culture for bacteria. You know, and you're a great incubator because you stay at more or less one temperature. And you're pretty moist. So bacteria really like you. Okay, we're going to look at the growth curve and then talk a little bit about what's going on here. What we have here is our log of viable organisms. Okay, so this is important, and we're also going to look at optical density. This is around, like, if you're in lab, if you weren't, about 600 nanometers. We're looking at scatter, and we're seeing viable. The spec method doesn't really work if you have a bunch of really dead cells because you don't really know what's going on. It becomes a problem when you start hitting stationary and death phase. But let's take a look at it. So in one case, people are culturing, they're figuring out how many cells are in here exactly, and then when they're doing it with a spectrophotometer, they're seeing what is in the culture tube and looking at how thick or how opaque or translucent the culture is. People do this quite a bit. Now, we start with the lag phase right here. Okay, the lag, let's just pay attention right now to the green curve. We'll talk about both of them in a second. In the lag phase, you've added culture, you've passed your culture. You've got an older culture, you think it's still alive. You want to pass it to a new tube. Maybe it's the same media. Great. If it's the same media, everything in that cell's ready to go. It's like, oh, I've been eating glucose and beef broth, and here's some other stuff. Good. I'm up, I'm ready to go. I jump in this stuff. Okay, I'm just going to start growing. Okay. So sometimes lag phase is next to nothing. Okay, there's not even sticker shock of being moved. As opposed to, let's just say that, you have your favorite culture of Pseudomonas, you're growing it on glucose and beef broth and that stuff. So now I want to see if the Pseudomonas is going to deal with petroleum or anthracene, some kind of pollutant that you're interested in. You put it in there might be a considerable lag phase, because it says, well, all right, I'm starving from carbon, I gotta get this stuff, but I'm not ramped up, I don't have those oxidases and stuff ready to go. So there might be a ramp-up time where ribosomes have to transcribe a messenger, and messenger has to go into protein and so forth. So that's what's going on in the lag phase. Okay, it can be next to nothing if you're just transferring. Could be a little bit longer. You can pay attention to that. That tells us something about physiology. Okay, exponentials are the next. So lag is kind of where it's flat in the beginning. Okay, let's just stay on the green curve here. Now exponential, we've been through that. That's where the cells are actively growing. If you have only viable cells, they're all doing binary fission. In our example, every half hour, I got twice as many of us. Okay, twice as many, you get it, okay, and it's going up. Now, in a batch culture, what is happening? That's going to affect its exponential growth. Yeah, so you're eating up your nutrients, and you're swimming in your own waste. As fun as that sounds, it's not healthy. It's not healthy for you, it's not healthy for the bacteria. Okay, they don't have a choice. This is what we do to them. That's how sick this world is, right? Letting them run around in their own waste. Well, there you have it, and it's going to slow you down. So when you run out of food, okay, and you're in waste, you can't grow very well. And when you have your reproduction equaling the death, so here R is greater than 1. I mean, reproduction, that's what's going on. R, making more. Here, reproduction and death are about equal. Okay, you can still squeeze out some more bacteria, but some more are dying. Okay, hard to see sometimes with a turbidimeter, but you can measure that. This is the stationary phase. Okay, stationary because the curve is not going up, nor is it going down. It's plateauing. Okay, so know a little bit what's going on there. Then, if you keep things going longer, you're eventually going to have death being greater than R. D greater than R. So death is outdoing reproduction. Now, you can have a whole bunch of things that go into dormancy, and that requires a more stable. We're being general here, okay? Lots of things can happen in this phase. But eventually, I'll tell you, if you take your favorite culture, I don't care what one you're culturing, you leave it on the shelf and you're on a slant or in culture, you can pass it for a while. All you need is one cell to get going, but if you wait too long, a month or something, most of your clinical cultures, they're going like, that's it, I'm out of here. Now, there's a bunch of ways you can trick cells, but we're just talking in general. So now we get the idea. We not only know what we're looking at time and number, and we know phases of this curve, and it means something to us biologically. So you should be happy about that. Now, we want to consider these two curves and what's going on here, and that is viable count versus turbidity. Now, you got to realize this will actually come over here and hit this too. They're starting in the middle of the curve, so to make a point. But the point is that if you're just going to measure cells in solution, you're going to probably overestimate things a little bit at the low end of just using light. Light's going to go through a light path. It's going to even though you subtract your control, it's going to try to show you something, give you some kind of reading, and you got to just take that in faith. Exponential, okay, still a little bit of an overestimation, but pretty good at estimating it. And then a little bit of underestimation as you go through here, as it becomes more dense, especially with time, it becomes less capable of seeing differences. Okay, and that's just a light path thing. We did this in lab. A lot of people are asking me about that. That's it. You're scattering light. You're not reading a specific wavelength. as like you would with maybe the Lowry assay or a protein assay or something you might do. Okay, I think that's enough on that. Bacterial growth curve. This is just an animation. If you want to play it, it pretty much goes over that. I don't think we need to. I don't think I need to watch you watch this, and you probably got the idea anyway. So let's talk about continuous culture, chemostats. Okay, you go to work for an industry. You're a microbiologist. You've got to make pretty good money. Okay, it's not, because you're combining chemistry, engineering, and biology. It's kind of a sweet gig. All right, you might be trying to figure out. I tell you what, one of the biggest, still lucrative, is trying to figure out waste management. The city of Chicago is fighting tooth and nail, having any kind of phosphorus limit of phosphorus that can be dumped into the rivers of Illinois. It's not that Chicago's a bad place. I mean, it's a city, it's got good and bad, whatever. But it's that every time you're trying to reduce phosphorus, you're trying to reduce the amount of coliforms, any reduction you're doing from how many millions of people taking going to the bathroom every day, that costs millions, billions of dollars. It's a big deal. People are constantly trying to figure out sewage treatment and making it more efficient. That's a continuous culture. Sewage treatment plants. You're putting in, influent. Right now, everyone gets up. What's the first thing people do? Boom. That's all going there. You have storms to deal with, and then you're trying to move something out. In between, you're trying to do these really big cultures and really big tanks. We're idealizing it here, but you get the idea. Continuous culture. So it's an open system. Stuff is coming in. You're letting stuff out. Chemostats, most common type of continuous culture. There's some other names for those. You might see biostat, things like that. But anyway, both the growth rate and population density of the culture be controlled independently yet simultaneously. So if you want to go through, let's just say you have this culture and you're growing things, you have a headspace, that's just kind of to allow for difference of effluent is coming in, influent, excuse me, and effluent. Dilution rate at which the fresh medium is pumped in, spent medium is going to affect then growth, and then the concentration of a limiting nutrient can also dictate how much growth is going on inside of it. We talked about Liebig's law of the minimum. You can use the minimum thing. If you're missing one thing from growth, it's still going to limit you. I mean, it doesn't matter. So you can maybe pump in a lot of glucose or whatever, post-toasties, whatever carbon you're using. But if your culture is limiting on iron or something, you can still control it. Okay. So really, you in the chemostat, you move up through this thing once you're ramping it up, and then you can kind of just maintain the growth on it. That's the neat thing about it. Now, you don't only have to do this for sewage treatment. I say that because we all have some idea of that. You could do this for the production of a certain compound. Let's just say you're trying to grow a lot of bacterial culture that's growing a certain byproduct that you want for the industry. Then you can continue to grow it at a high yield, take off the effluent, and distill out your material, and keep production really high also, and minimize contaminating materials. So, too high of a dilution rate, the organisms will get washed out. Too low dilution rate, and you may starve them out. So what you would do here when you're setting up your chemostat is quite literally you would be doing kind of biopsies and checking out, adjusting your dilution rate and then the concentration of nutrients. That's how that's set up. It's like getting a car engine running. You get it running at first, then you want to tune it up. That's what you're doing, is tuning it to the next level. So. Okay, let's talk a little bit about how we measure things. We know we can measure cells through culturing. Okay, I think I have a slide on that yet, but I think we've been through that. You can measure get an idea of numbers, if you have some way of first calibrating it, but you can do it with light. Here's a counting cell. Maybe some of you have seen this, counting blood cells. I don't know if they do this here anymore, but we have counting cells around. You can actually put a drop of culture on a cell. There's a very small grid. This is the size of a microscope slide. They have these little grids in here so you can count the cells. For us today in lab, we're going to do a version of this fluorometrically. Why? Because our eyes can see fluorescence 10 to 100 times better than we can see light in contrast. But anyway, I'm not concerned about this. If you start doing this, you would know the volume that you can put on the cover slip. It's actually calibrated. And then you know how much of the volume is over how much of an area. So then you can get your counts and back calculate. You can take area, convert it to volume, and then you have a number per volume. So people can do counts when counts are necessary. Okay, this one microscope counts. Why do I have a laser in there? Oh, I have a laser in there because it's from a confocal example. But either way, in flow cytometry, we can do this now. You can have a flow of cells coming out of a cell, and if you have things set up with different labels, you can count different cells coming through using different spectra off of lasers. So the big take-home message is like, oh, that's a lot. Flow cytometry, you can count cells very rapidly and with sensitivity using laser technology, I think is what I want you to know there. Do we have a flow cytometer here now? I heard somebody, one of the professors had one, but I never confirmed it. Does anyone know? Maybe Johnson might have one. Anyone work for Johnson? Anyone know him? Venom, yeah, I saw the worksheets. Venom. Okay. Well, anyway, I got to find out about that. Okay, well, we plate count. We know that we can essentially. There's different methods. You can put a known volume. We can spread things out. Maybe it's too much to begin with. We want to dilute it out many different ways. Spread plating is a way to do it. We can pour plate. But anyway, we can count cells. We can count colonies. Why is counting colonies going to help me to know about bacterial cells? Each colony is 1,000,000 billion cells. That doesn't seem to make too much sense. What's the connection I have to have there, or the assumption? There's a pure colonies, but how can I say that? How can I convert 100 million billion cells here to counting anything? Yeah.
One cell gives rise to a colony.
That's the assumption. Yeah, one cell per colony. So you're starting out. Yeah, in the beginning you can't see it. You can't see those bacteria. I mean, with your eye. But the assumption is that one little cell fell here, and they're going, "Wow, this is great! I fell into a plate of food." And they start. Growing and growing and growing from the inside out. So like a tumor, the inside is the oldest, the most degraded. It doesn't care. We just want to see a slimy little wad. Now, 2 cells fell here and they started to grow. We don't know. We have to assume. So we call them colony-forming units because we like to be conservative. You couldn't tell if 2 of the same creatures fell into the same spot. Okay. There's different methods of doing pour plate and spread plate. One is just spreading across the surface. Pour plate is mixing in with the agar. If you're in the lab, your old hands at that. If not, it's here for you to review. One thing you have to do is do dilutions, and that's all this is saying. You don't know. Someone hands you a culture. I mean, maybe you're tuned into a higher plane. I'm not. I just say, well, I'm just going to set up a bunch of dilutions, or I spread plate at a bunch of different volumes and try to decide where I'm at. Once I know the system, I've been doing Lake Springfield for 25 years. I can pretty much guess what to give students, and usually I'm pretty close. But things do change. But you get to know your system, you can do more. We've talked about turbidity. We're talking about light scatter. This is at 540. I'd be doing it at 600. Some people vary on that, it really depends, but, you know, you're saying that a cell will scatter light X amount, and more cells will scatter more light in a linear fashion until you reach quenching. And that's, I think we've kind of went through that quite a bit. We grow at different rates, no, we know that. The Great Plate Count Anomaly, have I talked about this yet? We're gonna do it in lab, so this is great, we're gonna do it in lab, if not, and we'll talk about it here. What is an anomaly? Now, this is something like, you know, this is a fairly common word, so it's not just like, oh, that's so specific, I would never know, but if you have anomalous results, or if the meteorite hit and all of a sudden, you know, what you thought is now an anomaly, what is that word? Who is Google? Who is Wiki? Who is Google? Give us a definition of anomalous. Yeah, there you go, that's what I want. This is the, you know, we're in the next century, we gotta use technology.
Is that something that deviates from what is standard, normal, or expected?
Something that deviates from what is expected, that's good, or standard or normal and it's expected. So here is a deal, okay, Konopka in the 1980s from Purdue, he makes a big deal of it. You go in the literature, people are starting to notice it in the 60s. People are saying, wow, we're getting a handle on disease. This is kind of interesting. When we look in nature, how much is out there? And microbial ecology, you know, is really about that old. You know, in the 80s, you know, we're only talking about a few decades here. People are starting to say, well, what is out there? And then they took the ecology into you. They're going, well, now let's see what kind of microbial ecology lives in people, and that's the human microbiome. Never would have gotten there without people like Woese and Pace and some of the people here in Illinois that figured it out and did all this great stuff. So, what is going on with the anomaly? Well, at that point in time, people were able to culture bacteria. They were able to do that back to Robert Koch's time, probably before, okay? And people were able, they're really doing a lot of counting. The very thing we're gonna do today in lab, where you take fluorescent bacteria and count the number out here. So this is a numbers game. It's not the species game. Okay, the stakes are a little higher now because we can sequence everything. So we're in the numbers game. So here's the deal. They're saying, okay, culturing, how much of the world, microbial world, do I understand just by growing stuff on plates? We've been doing it for centuries now. What are we really seeing compared to what's out there? So what they did is they went through, I mean, there's different ways of doing it, but quite literally what we were doing in the lab, they went through and they did a bunch of smears of water samples. They took soil and they vibrated off the bacteria and they plated those. They did all these counts. They tried different media and stuff, and they come up with a number. For us, what Lake Springfield would we have? About somewhere between 10,000 to 100,000 bacteria per mil, if I remember right, that we could culture. That's not bad. Okay, if you guys, it may still be up on the bulletin board. I did a publication, I looked at the literature all across to see if people were getting it. They get that. Okay, so about 10,000 bacteria or so can be cultured out of a mil of water, soil. Sometimes you get a little bit more, depends on the organic content of the soil. Now, well, how many are there? Okay, how many are in the soil? So they went and they did this DAPI stain. And how many bacteria are in a mil of Lake Springfield water? We're going to answer it today, but what do you think? More than been cultured? Yeah, probably. About 1 million. Okay, 1 million to 10 million. You go, oh wow, that's a lot. Well, you know, the orders of magnitude of bacteria is insane. So 1 million to 10 million in 20 drops of water. Okay, and you have about, and you go, well, boy, we're really underestimating stuff doing culturing. This fact, or this anomaly, set back microbiology, in my opinion. They worked out that 1% or less of the bacteria that exist in nature can be cultured. It's roughly about the same for you. you have bacteria in there that we can't grow on plates and stuff. So that's a figure you should write down, and let's see, we've talked about it already. 1% or less. Okay. Some places you can get a little bit higher. You know, if you have a bloom, you know, a bunch of ducks just landed, maybe you'll be lucky and grow a bunch of stuff. But about 1% or less. So this set off an explosion of people who say, well, that's why we have to do molecular work. You know, we'll never figure stuff out with culturing. Okay, and to some extent they're right. But now, when you go in to do molecular, you go in, you grab the soil sample, right? What you do, you need the DNA. Okay, you got the DNA, you sequence it, you do this, you do that with it, okay. Now, I got the answer. This thing is Bacteroides species, blah, 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 blah. Now what? I got a name on a paper, I don't have an organism. Okay, I mean, I ripped, it was summer in that soil sample, I ripped that cell apart and grabbed that. Now, you can go back, you know, and you find the messenger maybe, you can do some stuff. So molecular stuff is cool, don't get me wrong, love it, do it all the time, big fan. But culturing still is where things are at. In Central Park alone, my student, Mike Rossetto, tennis team guy, big athlete also, he found 100 new species through culturing in a unique way that Jansen and his group were coming from Australia. They had new ideas. We could do the same thing. We could go culture and find new stuff. Okay. So, first off, the Great Plate Count Anomaly, so I don't get too sidetracked, it's the difference between what you can grow, the number of bacteria, not in species, just the number, I can grow on a plate versus what's in the sample. Okay, so that's it. I will ask you on the test, quite likely, if I don't forget, I will want to ask you, don't forget, you know, what is the Great Plate Count Anomaly and what are the implications? So that's why I'm rambling a little bit to give you some implications. So we've talked about something, the molecular versus culture approach, okay? But the Great Plate Count Anomaly is somewhat vexed. And you would know why if I let you think about it. I'll save you a little time and I'll tell you. In that total count, are all those cells alive? Could those cells be cultured if you really even wanted to? No. Some of those cells are dead, they're just waiting to lyse, but they may light up if the DNA is intact. Okay, some of those cells may be culturable, they may be viable, but non-culturable. Rita Caldwell shows that with Vibrio cholera. We'll talk about that in a little bit. That cholera can be viable and you can't culture. You miss it if you're a clinical biologist. So they can be viable, but you can't grow them on plates. And the last thing is, maybe we're not thinking enough about how to grow stuff. You know, what you do in the lab is you add nutrient broth. You add tons of nutrients and carbon. Okay, you're setting up a copiotrophic culture. What's copious? Cornucopia. Horn of plenty. You are throwing in, it's like throwing you in a room full of whatever you like to eat, pizzas, whatever. You would do okay if you didn't smother, but you would do all right. You can get respiration going so fast that you actually burn up some of these cells that exist in nature, and it's kind of like the lone wolves of cells. They're kind of getting a little bit of food here and kind of hanging out kind of stuff. So they think that and they've done a lot more stuff, or just doing things like laying a membrane on the beach and letting things culture and pulling it out. But people are getting more creative about it because they're thinking about microbes as organisms and not just stuff that comes in, creates pus, and degrades my flesh. You know, that's okay. Now, again, we're moving through. We got past germs. We got into bioremediation. Now we have to think about, well, if we really want to learn, I hate putting those pandas into the zoo, but you know, I gotta learn something about those pandas before they go extinct. It's to that level because of the amount of information we can learn from it. Molecular, cool, learn a lot, but you don't have the specimen in most cases. All right, so hopefully sorry about going on about that, but it's a big deal and it represents, again, another perspective of how we change looking at microbes on Earth, and that's going to change in your lifetime as they understand the human microbiome with you more. You are in that next change. So, is my prediction, I think I'm right. In fact, I'm never wrong. Except when I am. Okay, so let's talk about growth. Now we get an idea. There is a relationship, generally a positive relationship, between increasing temperature and growth rate. Okay, and here we have temperature increasing left to right, and we have growth increasing from down to up on the y-axis. Okay, well, we all know how to read graphs. What's going on though? At some point this thing drops off. Yes, this is a growth curve based on temperature now. When we looked at it before, we were maintaining temperature. Now we're messing with variables. So, in this bacteria, if we grow it, here it's going to grow a little slower, probably like the bacteria on my leftovers in my refrigerator right now, but if I put it on the counter, they're going to grow faster and go faster. At some point it drops off severely. Okay, this is the optimum temperature, and here we have what's going on. You're going by Michaelis-Menten kinetics. As you increase temperature, the enzyme is increasing, okay? But you get to some point where the enzyme will denature for that organism, and growth is no longer possible. So you get to that point of where those hydrogen bonds can't take it anymore, and it becomes ineffective. Down here, this is important for adaptation. You get too low, and that membrane becomes really hard. Remember, it's a lipid. You know, lipids are, maybe it's nice and lipid bilayer at 35 degrees. You put it down at a low temperature, it starts to gel up, and you just can't get stuff in or out of the cell. Okay, this is stuff you kind of know, but we're putting it together. I don't think we're going, oh, I never heard of that before, but let's look at it a little bit more. Oh, the cardinal temperature, this is what I was looking for you. Cardinal temperature, minimum, maximum, optimum. They call those the cardinal temperatures, okay. Because those are the most important ones. Now, we can partition bacteria, the group of bacteria, the group of archaea, by their growth rates. It's one of the characteristics we can look at, okay? Not all bacteria, even though they live in many places as a domain, not all bacteria live everywhere, okay? I'd like you to have some kind of idea. Well, you shouldn't be familiar with these words, okay? It's just the prefix that is different. Here, we'll start with what we know. We're in this area, the mesophile, things that grow in us. Even though the optimum here is at 39, you'll pick up 10 of these different curves. They all have different cardinal temperatures. We're looking at things that are around body temperature-ish, 35, 39. That's it. Even though your cells love 35 to 37, they might go up to 39 or 40. They're not, then they're going to crash. So you try growing E. coli at 45, not going to do it. Well, it's nice and warm and toasty in there. Too toasty. Okay. And you can keep E. coli going, you know, even if refrigeration's at 5 degrees, 4 degrees, you can keep it going even when it's pretty cool. It's just not going to grow as fast. Great. Learn this. If you're working with a culture for your unknown and you want to slow it down, you know, incubate it at a little bit lower temperature. You can do that. You know, we use that to our advantage. Okay, so we get the idea there. Is there overlap? Yeah, they're being general. Psychrophilic bacteria. Neat. These guys will grow on glaciers. That's it. Not just, I'm hanging out on the glacier, I got blown here by some big hurricane, you know. No, they're sitting out there making more bacteria, you know, loving it. Optimum temperature of around 4 degrees. So you go like, well, wait a minute. I left my lasagna in my refrigerator and a bunch of fungus and stuff growing on it. Are those psychrophiles? Maybe, but you're not sure. And I'll tell you what. Those are psychotolerant. Okay, I mean, unless you can prove that they're actually reproducing, they love being in there and that they're very active. Usually we have psychotolerant things that grow in our refrigerator, and usually it's fungus. Although you can get plenty sick off of, you know, eat soup and milk and stuff around for too long a time. Those are usually psychotolerant. Okay, philic, you love it. Tolerant, well, you know, you put up with it, right? Kind of like so many things in life. Thermophiles, okay, hot. All right, 60 degrees is pretty hot. Okay, you would notice 60 degrees, it would start to burn you. Okay, I don't care that, there's many things here, but a lot of times you'll see names in there. A bacillus of the earth that's heated to, likes heat, okay. Thermococcus, okay, hypothermophiles. You say, well, why are there two here? Okay, well, there used to just be one thing. Hypothermophiles, those are the guys that are growing in the geysers. Okay, they're the ones when they drill, 7 miles beneath the Earth's crust, they find, stuff that loves heat. Not in a volcano, around a volcano. Can you believe it? Yeah, it happened. This one came in here, Pyrolobus fumari, had optimum 106. When this graph was made, I think that was probably it. Now we're at 121. Can you believe it? DNA doesn't even stick together at 94 degrees, and these guys are going, yeah, 121, I can do that. Yeah. It must be the Marines or something of the bacteria world. I don't know, they're pretty tough. So, and we'll talk about adaptations. Again, it's not magic. We can figure stuff out, we can learn from them. Just like we take the enzymes from bacteria that degrade cellulose, and they put it in your wash machine now to make your shirts whiter. You guys probably know about that stuff. We're doing all kinds of stuff, when we discover things, because we're really pretty interesting mammals if you look at us. What time is it? I have more time, right? All right, so here it is. Where do you find this? I don't think we're going to see anything on here. If you're growing, back to our original example, if you're trying to grow bacteria that are going to degrade oil, and you're going from nature, if you incubate them at 37, you might shut them right down. Pseudomonas flouris, Pseudomonas loves eating carpet, especially if it's small and healthy. They just love eating this stuff. Fluorescens, it doesn't like it. I mean, it might tolerate it, but if you put it at 40 degrees, it's not going to make it. It's one way of telling between different things. So anyway, in you, around you, your clinical ones, this is great. Growing in the trees, growing in that soup they call the Amazon River, they're going to do fine, okay. Extremophiles, you're looking at one place or the other, okay. Talked about that. Psychrophilic or hypothermic. Psychrophiles, cold temperature optima as opposed to psychotolerant. I think I've talked about that. It's in your notes. I think that's good for you now. Okay, some adaptations. Production of enzymes that function optimally in cold, features that may provide more flexibility. If you have more alpha helices, okay, you're building a structure that isn't quite as rigid. You want to keep it a little bit more fluid. Okay. Between oil and lard and so forth is the amount of double bonds and how to pack. Something that's simple. And that's a fatty acid, of course, but, making your proteins just a little bit different helps in the way how they fold together. More polar and less hydrophobic amino acids, okay, kind of keeping things moving apart. Fewer weaker bonds, so you don't have so much contraction in there. Decreased interactions in protein domains, so that you can have more fluid motion. Molecular transport function optimal at low temperatures due to modification of cytoplasmic membranes. You want to have more unsaturated fatty acids so that it's more oily. So when you cool it down, you can still get things through that. Stuff we don't talk about, we're usually at pretty much this kind of temperature. These guys, people are like, wow, they discover and they go like, how does it do that? And they start looking at these things. Thermophiles, talked about hypothermophiles. Okay, I think we've been through that. The DNA, when you start getting above 90, how many guys, any guys do PCR, polymerase chain reaction? One. Okay, well, if you meet Dr. Bonnicum, he will, it's a mantra with him. I've worked with the guy 25 years, and he still tells me every step of PCR, okay? But that's great. This is like, PCR and Gym are one, okay? That's the Zen there. But I tell you this because two things. The enzyme in PCR comes from a thermophilic bacteria, okay? Thermus aquaticus. And the guy who your book is named after, Tom Brock from University of Wisconsin, figured that out. You're doing DNA, you're making more DNA. You're not at 94 degrees and so forth. But the thing is with PCR, if we use your DNA or E. coli's DNA polymerase, it denatures. Thermus aquaticus is sitting around in a geyser, it's going like 94 degrees. I'm okay with that. I can do that. So Thermus aquaticus is a thermophile whose enzyme does not degrade at high temperatures, and we use it now for biotechnology. But the other thing that should blow your mind just slightly, if you know PCR, and if you don't, I'll tell you. In PCR, you have helical DNA, and you open it up, denature it, by putting it up to about 94 degrees. You take out the hydrogen bonds. Okay. Tac comes in, and then you make more DNA off that. When you get to these organisms that are living at this high temperature, you have to wonder to yourself, what stops the DNA from denaturing? And it turns out, just like the opposite, like the fish in the Antarctic have glycols, antifreeze to keep their blood from freezing. These guys have glycols and other helper proteins that stabilize the DNA. Why develop that? Why not just go find a cooler place to live? I don't know. The only thing I can tell you is that the Earth started out hot and smelly, and things had to develop in different places. If they started off today, maybe we wouldn't get things in that diversity, but when you look back 4 billion years, things were very, very different. Again, so trying to help you with ideas on diversity and why things the way that they are really helps in some of the essay questions. What do we have here? Okay, present. We already talked about that. We got a guy now. I don't know if they named it. They gave it a number last time I know, but your take-home message: animals, pretty narrow. I mean, you get up around 50 degrees. Some osteropods or little microcrustaceans. That's about it. They're probably just getting to the edge of the geyser and stuff. Plants, upper limit. No, plants don't do well. They dry up. Amongst other things. Many other things. Protozoans, fungi are more common. Okay, we get into bacteria. Some of the bacteria have extreme temperatures. Okay. With hypothermophiles, I just want to point out to you habitat. Some people think that if you go to a geyser, it's all the same, it's all the same temperature. There's gradients within things. Always look at nature when you can as what gradients are set up: pH, moisture, air, and heat. And these gradients define the niche of an organism. Okay. What's a niche? You want to take ecology? No one's in ecology. Some of you guys stay here after. Either you are reading or you're taking ecology, so don't lie to me. Take ecology. What's a niche?
Environment that something can live in.
Yeah, the place where it lives in. Okay, how's a niche defined? And we look at it from the back. I know that thing lives in that stump. That stump is its niche. Okay, a niche is defined as Hutchinson talks about it. And ask Amy. He's wrong or he's right. Great. Hutchinson said that every gradient helps to further define a niche. So a niche actually is a hypervolumetric situation. pH defines it, air, moisture, heat, all those different things. So then you start asking yourself, so here's a diversity thing again for building on that essay. How can we get so many different species of bacteria? Because they can dissect niches into so many different gradients. Okay, the optimum temperature, the optimum air, the optimum nutrient, all that kind of stuff can be different enough where the species can be very close in association, very close even genetically, and still be distinct. Okay, if you don't know what I'm talking about, just make a note. Look up Hutchinson. So G. Evelyn Hutchinson from Yale. Look up niche theory with Hutchinson. It'll come up for you. And just look at it, read it on Wiki. It'll take you 5 minutes. But that relating that to diversity is important. So that's what we talked about. All right. High temperatures, amino acid substitutions, sometimes help for, tolerant, in the heat. You increase ionic bonds, stronger bonds. The acidic, amino acids, the basic ones like, histidine and lysine, arginine, those are more, you expect more of those in there. And then this other thing, you produce like an antifreeze. You produce some of these materials that protect the DNA, okay, and can actually it doesn't work like dissipating. You can't dissipate the heat. There's nowhere to dissipate it to. It's not like the ethylene glycol that might be in the radiator of your car, because you're running it through wind, and it's dissipating heat. Here you're in the geyser. You're dealing with it. It's a different kind of situation. Cytoplasmic membranes, you want to have membranes with more saturated fatty acids. Okay, pH. Anybody here not know the pH scale? No one. Okay, so good, we'll move on. If you don't, look at that. I think we've had that since forever. What you want to do is talk about pH and what it has to do with microbes and culture. You'll find that different bacteria, just like they like different temperatures, grow at different pHs. And so, I'm going the wrong way. Here we go. So, what you want to do is talk about then what they are. If you have bacteria that live in acid, they're acidophiles. If they're living in alkaline, they're alkaphiles. Different bacteria will live that way. If you add a lot of bicarbonate to sewage effluent, is what they do to get rid of some bacteria, you'll actually get alkaphiles to grow. Osmolarity is another thing. Bacteria have tolerance to the amount of solute in solution. Of course, marine is different than fresh water. We have some that will actually go up to 5 normal sodium chloride. So these are our halophiles and halotolerance. Okay, so halophiles love it. Sodium chloride. They basically can displace sodium using potassium. So they have a little ionic pump in them. Okay. So 15 to 30% salt. So just think of having what's 100 mils? A half a cup of water. Okay. And adding 30 grams, like a good palmful of salt, and try drinking that, you would puke. Okay, most people would. So these guys, that works fine with them. Halotolerant, yeah, you can take it for a short period of time. You just can't exist in it, and then there's variations on that. It's really cool. We just started. You grow halophiles here. If you want to break halophiles open, you just spin them down and throw them in water. The water rushes in and pops them. You get the DNA really easily. But okay, air. We're just going through different growth gradients. Sort of people in lab, you've been through this, but let's just talk about it here. What this is showing, different bacteria, different pH, different salt, different all these things, can have different. They want air differently. We should talk about why. A little bit. First off, what are they? This is just showing tubes where the bacteria have been inoculated, and then they grow the best at certain depths based on oxygen. More oxygen here, no oxygen down there. Okay, and air is the oxygen source here. Okay, so if you grow just on top, okay, you're aerobic. Sometimes they call it, if you can only grow on top, obligate aerobes, okay, anaerobes. Facultative organisms, okay, you get the idea of what's going on here, what makes these facultative. These are just if they can metabolize glucose, what metabolism is going on there? What do you think? Or metabolisms? And the stuff we've talked about, so. Respiration, aerobic respiration. Aerobic respiration, you see the density, okay, a bunch of ATP being produced fermentation. Okay, down here we have air tolerant and we have microaerophilic. Why might cultures want to be microaerophilic? My theory is probably most cultures want to be microaerophilic, but we're not that far in making that generality. But what is the advantage of being microaerophilic when the more, is getting more oxygen necessarily better? What might be a factor in just reducing the amount of oxygen you're exposed to if you're a bacterium? And actually, for you guys, but you have protective things helping you out. What does oxygen do when you're metabolizing it? Some of it is a terminal electron acceptor, but what else happens?
You get the term just left my head.
Free radicals?
Yes.
See, I knew it was in her head. Free radicals, okay, and you get super oxygens, that kind of thing. What do those do to your cells if left unchecked? Yeah, they mutate, they mess up cells. So you have superoxide dismutase, you have catalase, you have things that break down peroxides and these oxides. And some of the bacteria do also. Okay, you ever have a cut, you put hydrogen peroxide, what happens? Bubbles, yeah, what are those bubbles? It's not CO2, it's not like you went, oh. It's oxygen, right? The peroxide is breaking down into water and oxygen. Okay, reducing your oxygen amount can help you. If you just have less oxygen, it's less oxygen to deal with. And to be microaerophilic, taking in just enough oxygen can help some bacteria not to have to deal with producing catalase and all those other enzymes. Aerotolerant, you grow all the way through here. What kind of organism, what part of glucose breakdown are organisms be doing, do you think? Not producing too much energy compared to these other guys. These are all, let's say, they're all cultured for the same amount of time. What part of glucose breakdown would account for aerotolerant anaerobes? Fermentation. Fermentation wouldn't do as well up there, but I think you're on the right track. Fermentation actually is pretty. Fermentation could work. Fermentation could work. Okay. Other. Just the production, just going through glycolysis. Okay. Sometimes some of the cells will truncate glycolysis, not go through and regenerate NAD and NADH, and get a few more ATPs that way. So if you just go through glycolysis, which is pure fermentation, you can do that. Strep does that. Okay, it grows well in your mouth. It loves your mouth, but it will be somewhat limited. Okay, you can culture without air. That's that. This is the slide on the different products, peroxide, free radicals. Talked about that already, so look that over. And that's the end of this. I think some people are falling asleep. Let's just start a new topic on Thursday. I'd like to get through advanced metabolism. Look at the slides if you can. If metabolism is kind of like not your thing, look at it. You'll feel more comfortable. I want to get into viruses before the test. Skip the genomics thing. I'll try to take the slides back down, but don't let that bother you. We're going to go stay on this idea of metabolism, and we're going to switch gears and get into viruses. That'll set us up really nicely for evolution and diversity in the second half of the course, or second 2/3. Okay, have a good day. We'll see some of you this afternoon. We're going to do a lot of staining this afternoon, so and it's my favorite lab because we would get you got to be good at your scope and try different things. So come ready to work, and we'll do that.